All right. Hey, everybody. So today we're just going to start out with a little introduction to our new chapter of study on genetics. And we are going to do that by first introducing you to someone who was known as the father of genetics. His name is Gregor Mendel. Now, Gregor Mendel, he was an Austrian monk who liked to garden. Specifically, he liked to work with pea plants in the garden. And while he was working with these peas, he noticed that there was a trend on which genes got passed down to future generations. And that trend is called inheritance or heredity. So when you are inheriting a trait, it just means that you're getting your genes, you're getting your traits from your parent generation or from previous generations. So when he was working with the peas, he started with two pea plants. One that had a round-shaped pea, the other had wrinkle-shaped peas. When he fertilized those two and produced a new generation of pea plants, he noticed that, for whatever the reason, all of that second generation of pea plants had round peas. There were no wrinkled peas at all. But then he took two plants from this first generation here, crossed those, fertilized those pea plants, and then he came to realize that the wrinkled pea, or the wrinkle shaped for the peas, came back. So that wrinkled gene never really disappeared. It just was overshadowed by this round gene, or the trait, the version for this round pea plant. And that's because we know that genes come in pairs. Again, we talked about this when we learned about meiosis. Yeah, you get one version of each of your genes from mom and one version of each of your genes from dad. And sometimes you display that trait from mom, sometimes you display that trait from dad. And we're going to learn a little bit about why that is. Before we get into that, however, you should be aware that any version of the trait, okay, in that past example, okay, you had round peas and wrinkled peas. The versions of those traits are called alleles. So you would have a round pea, you would have an allele for a round pea, you'd also have an allele for the wrinkled version of that trait, for the wrinkled pea. And that's because there are certain alleles or certain versions of the traits that are considered to be dominant, and those are symbolized by a capital letter. And there are other versions of the trait that are recessive. Okay? And for terms of our class, these are really the two main types of traits that we are going to discuss. A dominant allele, which again is represented by a capital letter, and a recessive allele, which is another version of that trait, which is represented by a lowercase letter. And that brings us to a few more new vocabulary terms: homozygous and heterozygous. So again, I know they sound similar, just like most other vocabulary words that we will see throughout the year in our biology class, but unfortunately, their meanings are fairly different. So a homozygous gene, and again, don't forget, right? You get one version of each gene from each parent. So one from mom, one from dad. That's why you'll see genes usually displayed in pairs. Okay, you have one allele, one version of the trait, and then another. But again, homozygous genes are showing you genes where the alleles are both the same. So if we're using the letter B to represent a trait, let's just say, for the sake of argument here, that we're looking at eye color. All right. So the big B represents a brown eye color. Little b represents a blue eye color. So a homozygous gene that would be for brown eyes would be two big Bs, right? Because you get one big B, one allele from dad, and then you get one allele from mom, giving you brown eyes. Or you might see two little b's. And remember, the lowercase letters are representing a recessive gene. So in this case, okay, we said the lowercase b represents a blue eye color. So blue eyes would be a recessive version of the trait. And that would be represented by two little b's. Now, a heterozygous gene is shown with both letters. So when you see that term heterozygous, think different, right? You're going to see two different versions of that trait. So in the genotype or the genes that we get from mom and dad, one might be capital B, one might be a lowercase b. If that's the case, this is heterozygous. Now, and again, I'll repeat this throughout the next couple of weeks. Anytime you get a genotype, which is what we're looking at here, where there's two letters together, if there is a capital letter representing the dominant trait or the dominant version of that gene, the dominant allele, that's what we are going to see. So if you remember, we said capital B represents brown eyes, lowercase b would represent blue eyes. So in the case of this heterozygous gene where we have a capital B and a lowercase b, okay, the dominant allele and the recessive allele are both present, you're actually only going to see the dominant version of that gene. So if somebody was heterozygous for eye color and that this was their genotype, okay, since they have that dominant allele, that capital B there, this person would have brown eyes. The only way that somebody would have blue eyes is if their genotype or their genes coded for the two recessive traits, the little b, little b. And here we go, genotype and phenotype. So as I was mentioning before, okay, genotype is, we're really using these letters. We're using the versions of the trait to describe it. So your possibilities of a genotype would be two dominant alleles, so two capital letters, two lowercase letters or two recessive alleles, which these are both homozygous because, again, it's the same letter. Or your genotype could be heterozygous, which is capital, lowercase. So heterozygous, and again, this would still show the dominant version of the trait, or its phenotype would be the dominant version. Okay, so phenotype is showing you telling you the observable characteristics that you would see. So think of it as like the physical appearance. So the way I try to sort of differentiate these two is genotype is talking about the genes. So we would look at the letters that we have. Phenotype, think of physical appearance. So what would we physically see based off of the genotype? Okay, so genotype, think about the genes, right? The different alleles. Phenotype, think about the physical appearance. What would we see? So we know that in meiosis, right, we end up with 4 genetically different cells. So again, when we get one version from mom, one version from dad, at the end of meiosis, okay, after that cell splits after the cell splits twice, creating 4 genetically different cells, each version of the trait, each new cell, each new cell would have a different version of that trait depending on the genes that you have. So in this case, right, the genotype in the cell we start out with is big B, little b, so the dominant version and the recessive version. And then when that splits through meiosis, you will then end up with one cell which would have the dominant trait or the dominant allele, or you would have a cell with a recessive trait, the lowercase letter, the recessive allele. And there's your example. All right. So what we can do to really give us a better idea as to what we can expect for future generations to show as far as their genes and what genes they will likely inherit from the parent generation, we do something called a cross. You might have also heard of Punnett squares, maybe like 7th grade-ish. But Punnett squares are an easy way for us to really identify what are the possible outcomes of these different traits in the offspring that are being created. So we're really just going to focus on a monohybrid cross, which is just one version of each trait, not dihybrid. So don't worry about the dihybrid cross. You're going to see what monohybrid looks like in a second. In this Punnett square, yeah! So a Punnett square, as I had mentioned in that previous slide, is just a way to predict the possible outcomes for the genes of the offspring of future, of future generations. So in this example, and we'll have plenty of practice with these moving forward, but in this example, one parent has a genotype that is heterozygous, big B, little b, and the other parent has a heterozygous genotype, big B, little b. And if you were curious, because I know you all are, the phenotype, the physical characteristic that these parents would have would be the dominant trait. So again, if we're still talking eye color here, we said before that brown eyes would be represented by a capital B because that's a dominant version of the eye color trait. So each of these parents would be considered to have brown eyes. So the way you do a Punnett square, I'll just breeze through it quickly, is you sort of fill in the parents on the top, and then you fill in these empty boxes. So you would bring this version of the trait down all the way for both boxes, this version of the trait down all the way for both boxes here, and then same thing across. And it's okay if that doesn't make any sense, because as we practice it, I think it will be very clear and you guys will be Punnett square experts. But basically, this is telling us that there's a possibility that the offspring could be homozygous dominant for eye color, which is the big B, big B. There's also a possibility of having a heterozygous offspring. So again, big B, little b, showing you heterozygous because it's two different alleles. And there's also a slight possibility to have a homozygous recessive offspring, which has both lowercase letters or both recessive alleles of this trait. So in that case, right, all three of these offspring would have brown eyes, but there's a 25% chance that there would be an offspring with blue eyes. Again, if we're talking about eye color. So that's a little bit of an intro to genetics. Oh, there's a phenotype. I just kind of said it already. But again, 75% would have brown eyes because, again, you have the capital B and 3 out of the 4 squares here, and then only 25% would be possible for blue eyes. But as I was saying, before I was interrupted by that little piece of information that I forgot about there, that's a little intro to genetics. Don't let this new vocabulary overwhelm you. I promise it will make a ton of sense by the time we finish with this chapter. It's all new, so it's totally okay. So hopefully you learned a little bit of something, and can't wait to see you guys soon. All right. See ya.